All right, so picking up where we left off, talked about the end of the 5 I's, right? We talked about the 5 I procedures that take place in most microbiology laboratories in order to identify microorganisms. Now we're going to dive a little bit more into that inspection eye, right? Half of that is looking at it with just our naked eyes, but another big part of that is looking at it under a microscope, right? These are microscopic organisms. We put a lot of effort and importance into seeing what organisms look like underneath a microscope. So we're briefly going to talk about microscopy and how you use a microscope, what a microscope does, and some advantages and disadvantages to certain types of microscopy. And again, there are going to be quite a few slides in this section that I skipped. So if I just put an X through it, we don't have to learn it. We're not going to talk about it. I decided it was too much detail. Okay, so microscopy, this is just an example of what some bacteria look like under a microscope. Skilled microbiologists a lot of times can diagnose infections based on just how it looks under a microscope. There are a lot of organisms that have very specific shapes and sizes and staining, and so they look a particular way underneath a microscope. And there's a lot of information that you can get by simply looking at a bacteria underneath a microscope. Okay, and this is just kind of a reminder. You don't have to memorize the numbers on here, but this is just giving you a relative size chart of how big things are relative to each other. So the human eye has about a range down to, again, about 1 millimeter. Okay. Sometimes a little less, right? We can see things that are a little bit less than a millimeter. And sometimes it depends on like the lighting. Like right now in this room, most of us can't see any dust floating around, like in front of our eyes. There's no dust motes. But sometimes if you look just right, like where the light is shining from the projector, you can see little things floating in front of it, right? The things floating in front of it are all over the room. It's just that right there, the light is shining just right. Or when the windows come, or the sunshine comes in through your windows at certain times of day, you can see the dust bunnies better, right? Things like that. But the limitation of the human eye is about 1 millimeter. And there are some microscopic structures that we can see, again, if the lighting is just right. But most cells are not going to be visible by the human eye. So Giardia lamblia and red blood cells, these right here are eukaryotic cells. Giardia lamblia, we'll talk about him later if you don't know who he is, but it's a protozoan. It's a parasite that causes what's called traveler's diarrhea. It's a particularly heinous version of like water poisoning. And then a red blood cell is what the cells inside of our body that carry our red blood cells, so that's about the size of a human cell. And then yeast tend to be about 10 micrometers. And then down here we have our prokaryotes, which are things like bacteria and archaea, right? E. coli bacteria, mycoplasma bacteria, and then we would also have archaea in there, things like that. And then below that are viruses. So it's important to note that there are a lot of different types of microscopes. And the main microscope that we are going to be talking about and are the most common in human microbiology, or in microbiology and in specifically college, is a light microscope. So the microscopes that you have seen, most likely, right, the regular kind of microscope that you've probably used in high school biology class or whatever else, or if you've been to micro lab for this class, those are the kind of microscopes that you are probably familiar with. And they are called light microscopes. Light microscopes are the most common type. They're called light microscopes because they use light. But it's important to note that the range of a light microscope It can view everything from eukaryotic cells down to most bacteria. Some bacteria are a fair bit smaller than that, but most average bacteria are going to be visible with a light microscope. However, viruses are not. Viruses can only be viewed, they're much too small, they're about 100 times smaller than most bacteria. Viruses can only be viewed through something called an electron microscope. Okay. And it's a microscope that uses special electrons to bounce off the organism and makes it able for you to see it. These are really, really expensive, as you can see in this picture over here. A lot of times electron microscopes are very, very large. They take up huge amounts of space. A lot of times they sit on the floor. They're so big. Okay. So viruses are not going to be viewable without a special microscope. Bacteria, eukaryotes, yeast, things like that will be. Anything smaller will not. Okay. All we're really going to talk about on this slide, this is just a basic picture of an average light microscope. This is a light microscope. This is the one that you will become most familiar with and the one we will talk about the most. Electron microscopes are much more rare and not commonly used, especially by students. And basically the way that a light microscope works is the light source is down here at the bottom. And the sample will sit right here on the stage. And then the person trying to view something will look through the lenses, which are up here. So the lenses are the part where your eyeballs look in. The stage is that middle part where the sample will sit, and the sample is usually put on a glass slide, but it will sit right there. And then the light comes up from the bottom. And so the way that this microscope works is the light comes up from the bottom and it shines through our sample and into the person's eyes. And the light shining through the bottom, through the sample, and then the light bending through those lenses is what makes the image appear bigger. Okay? That's all we have to understand for this class. Obviously, microscopy is a whole science. We could talk about light bending and refraction and all kinds of stuff for a really long time, but we're not going to talk about that in this class. So this is a basic light microscope. Again, light microscopes are only going to allow you to see things that are about the size of a bacteria and larger. Viruses and proteins, things like that, are much smaller and are not visible through this microscope. Okay. The major purpose, there are a couple of different properties of microscopes that make them useful for visualizing bacteria. The first characteristic is magnification. Basically, they make it bigger. They make an image, oh, I don't need to write it. It's the extent to which lenses make an image appear larger. That's how we are able to see the bacteria, because the light traveling through the sample and through those lenses are basically mirrors. So through those lenses, they bend light and reflect light in such a way that the image appears bigger. Okay, we're not going to talk about total magnification. We're skipping it. You will learn about all of that in lab. If you're taking lab, they'll cover it there. Okay. And so this is just a picture of what certain types of cells can look like under different magnifications. So a lot of microscopes have multiple stages of magnification. The biggest ones are, we have 3 major levels of magnification. 10x means that it appears 10 times larger. 40x is 40 times higher, or 40 times bigger. And 100x means 100 times bigger. Most microscopes have multiple levels of magnification through multiple lenses. okay? And this is just a visualization of what those microscope views would be under these different types of magnification. So this is under the 10x, this is under the 40x, and this is under 100x. And these pictures are all human blood. So those purple things there are red blood cells. So you can see under the 10x magnification, right, if you just look at blood with your naked eye, it just looks like red liquid, right? You can't see individual cells or anything like that. It just looks like a homogeneous red liquid. But if you put it under 10x magnification, you can start to see the individual cells. They're very, very clumped together still. You can't tell a lot of detail, but you can tell that there are individual little circles in there. You magnify it a little bit more, and you can start to see the edges of individual cells. If I had to count cells, I probably could, because I can see where each cell begins and ends. And I can also see different types of cells. So I can tell that like most of these, like all of these in here, all these purple things, these are red blood cells. But you might notice that there's a couple of things, like this one down here and this one up here, they look a little different. That's because those are white blood cells. So I can start to see some details. I might be able to identify some things based on that. And then this is what human cells look like under 100x magnification. You can see a lot more detail. I can count the individual structures inside these cells, right? So that's just a comparison of what different cell types will look like under 10x, 40x, and 100x magnification. 10 times bigger, 40 times bigger, and 100 times bigger. Now keep in mind, these are human red blood cells. Human red blood cells fall about there on our relative size chart. Bacteria are going to be quite a bit smaller. So bacteria are going to look a lot different. Bacteria under 100x magnification look more like these. Okay. So you can't expect bacterial cells to be as big as human cells, but this is just an example to show you how much that magnification increases with the different lenses. Okay. Do I want to talk about this? No, I don't. Okay, we're going to skip resolution. Resolution is another property that microscopes have. It has to do with the blurriness of an image when you make it bigger. Like if you've ever tried to zoom in like on your phone camera, at some point you zoom in and it's just blurry. Right? It keeps getting bigger, but you can't see anything. And that's what resolution is, but we're going to skip it. This is also resolution. I'm going to skip this too. And we are going to skip contrast. Okay. But what I do want to talk about is sample preparation. So when we are trying to look at samples underneath a microscope, there are two major ways to do that. One is through what we call a wet mount or a wet preparation, and the other one is through fixation and staining. Okay? And each one has pros and cons, and it's important that you understand the difference and why you would want to use one over the other. Okay, so a wet mount or a wet preparation is putting liquid sample directly on a glass slide and putting that glass slide underneath a microscope and looking at it. Again, if you're taking lab, I think that you may or may not do this where you just look at a drop of water underneath a slide. But it's just taking a little bit of liquid, putting it on a glass slide, and popping that underneath a microscope. This can be done in a lot of different situations. You can look at water and stuff like that. You can also look at urine or pus collected from a wound, right? Those are all liquids. You can look at blood. You can look at fluid from somebody's brain. Any kind of liquid, you can put it on a slide and stick it under a microscope directly. Now, the big pros of this is it's quick. And it's cheap. Okay. It takes literally 5 seconds to put a drop of something on a slide and stick it under a microscope. There's no preparation needed, there's no extra steps, there's no staining, there's no drying. None of the other things. It's quick, fast, and cheap. It is very useful for identifying the presence of things. So what I mean by that is it's a really good way to check to see if something is there. It is not going to be useful, and I'll write this down in a minute, for distinguishing details, okay? But it is very useful if you have a sample of somebody's cerebral spinal fluid, which is the fluid around their brain. That is not supposed to have anything in it. It is supposed to be, cerebral spinal fluid is supposed to be cleaner than water. It's supposed to look like pure water underneath a microscope. There's nothing supposed to be in it. But if I get a sample of somebody's cerebral spinal fluid and I put a drop of it on a slide and I look under a microscope and there's stuff in it, then I know that there's a problem already. So it's really, really good for identifying the presence of material. Is there a problem here or not? Okay. What it is not very good at, it is not useful for details or bacteria. So the other thing about, and then again, this is something you'll cover more in lab, but the other thing about wet prep samples is that they can only be viewed under the 40x magnification, under 10 and 40. You cannot go to 100x on a wet prep. And the reason for that is because the water takes up too much space and the lens will physically get stuck in the water. And you don't want that. So a wet prep or a wet mount can only be looked at under 40x. Bacteria require 100x magnification to really see anything. So this is not going to be useful for bacteria or distinguishing details. It is mostly good. It's a quick, cheap, and easy way to check for the presence of cells or not. Okay, so a really good example is this one right here. This is human urine. Now, again, human urine typically doesn't have very much in it, at least not much that can be seen under a microscope. There may be some cells, particularly if it's from a female. But there may be a few cells, but those are going to be skin cells. They're going to be external stuff. There may be some fibers if something from somebody's underwear got in there or whatever. But there's not typically like red blood cells or white blood cells. You're not supposed to have blood in your urine, things like that. And you're most certainly not supposed to have yeast or bacteria. Those things are indicative of an infection inside the bladder and urethra. So this sample right here is of human urine. And again, this is something that you have to be a little bit trained to look at. But this right here, that is yeast. And then all of these things over here, you can kind of see they're labeled. Those are white blood cells. And even an untrained person looking at that picture, is there nothing in that picture? No, there's stuff in it, right? So that's indicative that there is more of a problem than we want. And a lot of times that's enough. Getting a sample of urine or vaginal fluid in the emergency room is incredibly common. Somebody comes in with abdominal cramping or pain while they urinate and things like that, and you look at their urine under a microscope, and the person in the lab says, yeah, there's yeast in it. You call the doctor back and say, yeah, they have yeast in their urine. The doctor says, great, they have a yeast infection. Diagnosed, done. Okay, that took 3 minutes. No other testing was necessary. We didn't have to culture anything or identify anything or anything else. Everything was done just by identifying the presence of something underneath a microscope. Okay? So none of those extra steps were necessary. In this case, the wet prep was useful. Okay? But something I want to point out on this wet prep is you see these little tiny dots in the background? You can kind of see them floating around. Those are bacteria. Now, I don't know about you, but I can't tell anything about those bacteria. I can say that they're there, but I couldn't even begin to tell you what kind of bacteria that is. Is it causing a problem? Is it normal flora, right? Normal human vaginas have lots of bacteria in them. So is that normal bacteria? Abnormal bacteria? I don't know. So this type of specimen and preparation is not useful for certain applications. If I needed to identify the bacteria in this urine sample, this wet prep is not useful for that. Okay? So everything has a reason, there's a purpose for everything, and pros and cons. So again, pros of a wet mount, it's quick, fast, cheap, and it will identify the presence of certain things. It can tell you if there's cells, if there's yeast. You can even see if there are some bacteria, things like that. But it is not going to give you details. It is not going to help you identify anything. And it's not really useful for looking at bacteria. And it can only be looked at under the 40x maximum magnification. Now, fixation is our other way of preparing slides. And fixation is when a sample or a specimen is stuck to a slide using heat, and then it is stained, meaning that we apply different dyes to it to make it turn different colors. And there are lots of different dyes, and the purpose of each of these dyes is different. Each one looks at something specific. We'll talk about one specific slide or stain in just a second. But the pros of this are that it allows you, it can be looked at under the 100x magnification. So it is more detailed. So it can be viewed under the 100x magnification, which means that you can view it on a higher magnification and you can see more details. And then the stains also provide extra detail. Okay, so in this picture up here, this wet prep, there was no stain applied to this. Everything is the same color. Everything is clear. Okay, I only know what that is because I know what it is. I've spent a lot of time looking at it. But things are not different colors, right? Everything is the same color. Some things can get lost amongst the other things because they're all clear. But when you add stains, you can add different dyes that will stain different things, different colors. And that can allow you to tell more detail. The cons of this is that it takes longer. There are extra steps. It does require a little bit, not very much, but it does require a little bit of specialized training. You have to know how to apply the stains, how to fix something, what to look for. So it does take a little bit longer and requires a little bit more training. And then the other con is that the sample is killed. So something we didn't talk about when we talked about wet prep is because you are taking a sample and putting it directly on a slide, that slide is still alive when you do a wet prep. Now, sometimes that's useful, sometimes it's not. There are specific specific organisms that movement of that organism is incredibly important. A really good example of that is something called Trichomonas. Trichomonas is something we'll talk more about later, so I'm not writing down the name. This is just an example. But Trichomonas vaginalis, I'm sure you can guess where it grows. Trichomonas vaginalis is a vaginal sexually transmitted infection. It is a small little parasite. It's not a bacteria. It's a eukaryote that literally swims through the vaginal tract and the urethra, particularly in people who have vaginas. And you can diagnose somebody with trichomonas because you can literally see it swimming in their urine. You put it underneath a microscope and you can see those suckers swimming straight across the view slide. They're really easy to spot because they literally swim. Okay, so that movement makes it incredibly easy to see them, and it makes it incredibly easy to diagnose them. So in the case of a wet prep, I guess we can add another kind of pro up here. is that the organism is alive. So one of the cons of a fixation is that it kills the specimen. So if you are trying to diagnose something that has movement or movement is necessary, heat fixation is not going to be a good idea. So if you have a vaginal sample from the ER and somebody suspects that this patient has Trichomonas vaginalis, the best choice is a wet prep because vaginalis moves, and that's the easiest way to diagnose it. If we heat fix it and stain it, we're killing it, and it will no longer move because it's dead. Okay? So fixation, heat fixation and staining is most often used for looking at situations where we're looking for bacteria. Because again, this can be viewed under the 100x, the higher magnification. So it's really useful for looking at bacteria or any case where we need to see detail. Okay? So this sample right here it's either human urine or a vaginal sample. It's probably a vaginal sample. Okay? Now, these two samples come from very similar areas. In fact, the top one could be from a vaginal sample. It's hard to tell. It's either urine or vaginal. But those two specimens come from very similar areas. But notice that those are two completely different pictures. We see completely different things. The bottom one has a lot more detail. And these little tiny little scraps up here that I said were bacteria that you could barely see, especially from over there, are, when you view them after being stained at a much higher magnification, are now way more visible. So in this picture, you see all those little pink dots? All of these guys right here? Those are bacteria. And again, this is not something I expect you to be able to do, but that bacteria is so well defined, I can tell you a lot about it. I can tell you that that's a gram-negative cocci. It's a gram-negative cocci that exists in pairs. Again, I know you don't know what those words mean yet. We'll talk about it in a minute. But somebody who knows what they're looking for, what I can also tell you is that these bigger things right here, these are white blood cells. Specifically, they're called neutrophils, but they're white blood cells. And you can see, notice that all that bacteria, how much of it is in the white blood cells. Like this white blood cell up here, it's got tons and tons of bacteria infecting the inside of that cell. There's one organism that infects vaginal cells, particularly vaginal white blood cells, that is a gram-negative cocci that appears diploid. Okay, and that organism is gonorrhea. So this person has gonorrhea. I can tell that from just the microscope. Okay? But if I looked at this unstained and only at 40x, so if I tried to diagnose this person with gonorrhea without staining it, I wouldn't have any of that information. It would look like this. This tells me nothing, right? Look at the size difference. This is what a white blood cell looks like at 40x when it's unstained, and this is what a white blood cell looks like at 100x when it's stained. There's a lot more detail there. Okay? So those are our two major types of preparations. We have a wet prep, which is unstained, it's fast, quick, easy. It's good for the identification of the presence of organisms, but it can only be looked at 40x, and it doesn't show great detail. Fixation can be looked at 100x, shows a lot more detail, is useful for looking at bacteria, but it does take a little bit longer and does take some amount of training. And it does kill your sample. Okay? Now, like I said, there are lots of different stains that exist in the microbiological or microscopy world. We looked at this one back here, this kind of purple stain, right? This is a stain that's specifically used for red blood cells. And there's a lot of different stains that achieve different purposes. They have different reasons for being used when you're trying to accomplish different things. But by far, the most common stain used in microbiology, specifically clinical microbiology, is something called a Gram stain. And it's the only one that we're going to discuss in this class. You may talk about a few others in lab, but mostly you'll be talking about a Gram stain. It is by far the most important stain, most clinically useful. And the way a Gram stain works is it differentiates organisms based on their cell wall. Now we're going to talk about what a cell wall is and how the cell wall is different from a Gram positive and a Gram negative in the next chapter. But all you have to know for now is an organism that is Gram positive will appear purple. So this organism right here, that organism is purple. Okay? That is a Gram positive organism. Now, something I want to point out, I experienced this the last couple quarters, and I think it's just one of those things that thought processes, change with generations, not necessarily in a good way or a bad way. It's just we look at things differently when we're from different generations. And so I think it's kind of a thing of maybe the younger generation that I don't relate to as much. But when you're looking at Gram stains, your choices are pink or purple. Okay, sometimes the purple one will look a little bit more blue. Okay, sometimes it'll look a little bit lighter purple. But if you have two choices, it's pink or purple. If it's blue, pick the one that it's closer to, right? Blue is closer to purple, whichever one it looks closest to. I had lots of people last quarter in lab who were looked at a Gram stain, they're like, well, it looks blue, so that must mean it's negative. No, blue is not a choice, okay? It's pink and purple. Not an option. Blue is not an option. Pink or purple. So pink or purple is Gram positive, and Gram negative is pink. And I will use this little equal sign a lot. This means negative. Because if I was just to write a line, that doesn't mean anything to anybody. I know a negative sign or minus sign is usually just a line, but it's very common in most scientific fields that when you're typing out or writing out negative, you use a little 2 lines. Okay. So negative organisms are pink. Sometimes they look a little bit more red. I think that this one actually looks a little bit more red than pink, but it's in the pink family. Pink, red, maybe even a bit magenta. And here's some examples of a Gram positive and a Gram negative mixed together. So this slide has purple Gram positive organisms right next to pink Gram negative ones. Did I say that right? Purple Gram positives, pink Gram negatives. Okay, so you just got to memorize those colors for this class. And again, this is another one where it's really hard for me to overstate how important Gram staining is. Particularly because when bacteria are stained with a Gram stain, like it says up there, we're testing their different cell wall properties. And when we relate that to human health and human science, different bacteria with different cell walls relate to antibiotics differently. So medicine used to treat bacterial infections, which medicine you want to use is greatly dependent on what organism is growing, okay? So, and I will talk, and if you've taken my class before, I'm sorry you've heard this story a million times, but I tell it in all of my classes just because it's an easy story for me to tell because it's my story, and I don't have to worry about HIPAA violations and worrying about things from work and can I talk about it or can I not. And I think it provides a lot of context to what we're going to be talking about. So I'll bring up this story kind of a lot in relation to a lot of this stuff. But in 2020, I was in a car accident. I was in a really bad car accident. I was hit by a drunk driver. I broke my pelvis, my spine, both my legs, one of my arms. And one of the things I ended up with was a horrific infection in my leg. I had a really big abscess. It went all the way from my hip down to my knee. I had to be cut open. I had I want to say 10 surgeries because it kept getting reinfected. It was a whole thing. I was in the hospital for 7 months. And this happened as a side note, it happened in January of 2020. So I was still in the hospital in March of 2020, which we all know what happened in March of 2020. But it was a very long process. But the reason I bring it up was when I was originally in the hospital, they were dealing with all of my broken stuff. Got that fixed. I had a bunch of surgeries to bolt me back together. And then eventually I got sent home. Learned how to walk again, all that fun stuff. And they sent me home. Well, when I was home is when I developed an infection. I was at home and I had to go back to the emergency room with my leg was swelling up. I actually had pus coming out of my leg. The infection happened because of a seatbelt burn, right, where the seatbelt burned across my skin, which probably saved my life, but it ended up with an infection. But I had this huge abscess. I had a fever. I was really, really sick. I looked terrible. And so I go into the emergency room, and they take one look at me, and they say, yeah, you have a massive abscess in your leg. You're at high risk for developing sepsis, which is when the bacteria move into your bloodstream. It's incredibly dangerous and incredibly deadly. You can die from sepsis very, very fast. So when you have an abscess like that, the first priority is giving you medicine that's going to stop you from getting sicker, right? At that point, the abscess is extra, right? You got to go in order of severeness. Like, yeah, an abscess is terrible. It's not fun. We don't want to cut your leg off. That's not great. But you have to survive first. So the biggest thing is stopping the bacteria from spreading. Well, as we talked about in those first 5 I's, identification and getting the exact perfect name for this bacteria and finding out the perfect antibiotic takes days. It takes 3 or 4 days. When you have an abscess like that, you don't have time to sit in a hospital and wait for 4 days. So what a doctor wants to know in that situation is, what is the best antibiotic that I can give right now with the information that I have, right? Not the information I'm gonna get in 4 days. I want whatever information I can get right now to make the best decision I can make in this exact moment. And so what they did, and what they do, and is very common, is they stuck a swab into the pus that was coming out of my leg, swabbed it, and sent it down to the microbiology lab. It's kind of fun because I worked at St. Francis at the time, so all my friends were texting me and telling me what was in my sample, because I was working there at the time. But they put it on plates, they inoculate it, they incubate it, they do all that stuff to let it grow, but we need answers now. And so what they can do that takes 10 minutes is they can put it on a slide and stain it, and they can tell whether the organism is gram-positive or gram-negative. And that is going to cut our options in half. And it is going to direct the best possible outcome, right? And so finding out whether an organism is gram-positive or gram-negative is critically important, especially when it comes to healthcare, when decision-making time matters. And as you can tell by the fact that I'm standing here talking about it, I didn't die and my leg didn't get cut off. I have a wicked scar, and it took me a long time to get out of the hospital, but other than that, I'm relatively unharmed. But a lot of that is because they didn't wait. If they had waited 4 days, I probably would have died, which sounds really dramatic, but it's true. So again, it's really hard to overstate how important a gram-negative and a gram-positive stain can be. Again, I tell these stories because it's really important always to remember you're looking at a picture of something pink and purple on a slide. It's not about something being pink and purple. It's about a person on the other side of the slide. Okay, and it's always important to remember why we're doing that. Why do I care whether this is pink or purple? And the reason is because there's a patient that cares. There's a patient who could die if we don't get this answer correct. Okay? Not to be dramatic about it, but it's important. Okay? I like to just remind us all why we're doing this. This isn't just about learning dumb microbiology because your nursing department told you that you had to. Okay? There are important reasons for knowing all of this. Okay? And again, by the time that we get to the end of this class, you'll be sick of me talking about my own story. But like I said, it's an easy thing to talk about because it's mine, and I don't have to worry about HIPAA violations. Okay? And if you want to ask me questions, you can. I don't care. A lot of you can come to my office or ask me questions after class. Lots of people always have lots of questions. You can ask me questions after class if you want. I don't mind. All right, so that was chapter 2. Those are kind of the basic tools that we are going to use to talk about the different types of cells that we find. And that is the basis of what happens in a microbiology lab. All right, so now chapter 3 and 4, what we are going to be doing, we spent chapter 1 talking about the things that all cells share, right? The 4 things that all cells have to have: cytoplasm, ribosomes, membranes, and DNA. Those are the 4 things that all organisms have to have, all cells have to have. Now we're going to talk about some of the differences between bacteria, archaea, and eukaryotes. And then we're going to talk about viruses. So again, this is kind of an overview, a reminder of what we talked about before, that all cells have DNA, or genetic material, ribosomes, cytoplasm, and a cell membrane. If you don't remember what those do, go back a couple chapters. So those are the 4 things that all cells have, no exceptions. One of the things, there's no exceptions. However, we are going to be talking a lot about the difference between a prokaryotic organism and a eukaryotic organism. And they have a few major differences. The biggest one is that, can anybody tell me what it is? Because we already talked about it once. What do eukaryotes have that prokaryotes don't? A nucleus. Correct. Eukaryotes have a true nucleus, and prokaryotes have no nucleus. They have something that can be kind of similar. It's called a nucleolus, but it is not a nucleus. Okay? The other thing that kind of eukaryotes have that prokaryotes don't is very related to a nucleus and no nucleus, but it is that eukaryotes have what's called membrane-bound organelles. Okay. We're going to talk about what all those organelles are, but they basically have mini structures inside of their cells that are responsible for doing certain functions. Within a prokaryotic cell, everything is just free-flowing in the middle of the cell. There's nothing separated by a membrane. A eukaryotic cell I like to think of as a house, like one of those old-timey houses, not old-timey, but older types of houses that have a separate kitchen, right? Everything is completely walled off, and then the dining room has a door on it, and then the laundry room's got a door on it. Everything's got a door, everything is separated by walls. But now, that's what a eukaryotic cell is like. Everything has its own room. This is where protein synthesis happens, and this is where reproduction happens, and this is where this happens. Everything is separated by membranes into organelles. A prokaryote is more like an open concept house, right? Like the house, most of the houses being built today, where the dining room and the kitchen and the living room are all kind of one big room. Everything just happens in the middle. Okay? There's not really separation. Okay? So those are the main things, those are the 4 things that all cells have, and then those are the big differences between a prokaryote and a eukaryote. We are going to talk about a few more, but those are the big ones. Okay. So first, we're going to talk about bacteria. And then one thing to keep in mind, like as we go through this class, is that we are going to spend more time talking about bacteria than anything else. We're going to talk very briefly about archaea. We will talk about eukaryotes a little bit, but most of what we're studying here is microbiology of human health. The most significant microorganism in human health that's alive is bacteria. Okay, so we'll talk about bacteria more than anything else. And there's just another picture of the tree of life, just as a reminder. Remember that bacteria, that these 3 things, these are the domains. Bacteria, archaea, and eukarya. Those are the domains, but bacteria and archaea are prokaryotes, and eukarya are eukaryotes. Okay, so before we can talk about specific organisms and characteristics that they share, I do want to talk about what a bacterial shapes and arrangements. So something that is very common in bacteria is bacteria only come in a few shapes. Okay? Eukaryotic cells are not that way, and why that's true I'll talk about a little bit later. But eukaryotic cells, like even within your own body, you have cells of dozens and dozens and dozens of different shapes. Your heart cells are not the same shape as your intestine cells, which are not the same shape as your brain cells, right? And a lot of times the shape is really important to what it does. Brain cells have big, huge, long things in the middle. Those are your nerves, right? They form a very specific shape. But bacteria and archaea to some extent, but bacteria mostly have a few very specific shapes. And because there are so few shapes, again, we love to categorize stuff. We always want to put stuff in boxes, but a particular shape of bacteria can tell us a lot about what the organism is. And remember, the end goal with a lot of this is identification. What can I tell as quickly as I can, basically? And shape and arrangement are things that you can see underneath a microscope. Okay, so bacterial shape it's the shape of an individual shell. Shape of an individual cell. Okay, and we have specific words for specific shapes, which I'm going to go over in just a second. Another thing that's really important to note at this point is that all bacteria are unicellular. And what unicellular means, what does that prefix uni mean? One. Uni means one. Unicellular means one cell. That means the whole organism, the whole being, is one cell big. Okay? You are not unicellular. You are multicellular. You are made up of millions of cells that all work together to create one single organism. Bacteria, it is one cell per organism. Okay? So when we're talking about bacterial shape, or the shape of an individual cell, we are talking about the shape of an organism, what shape the organism is. Okay? And there are three major shapes. There are a few others, but you only have to remember these three. The shapes are listed up at the top. So our three shapes are going to be cocci. Cocci are basically circles, or spheres, however you want to call them. Okay, and all of these are cocci. They're circles. Oops. Bacilli is the second shape, and these are basically rods. They're elongated bacteria, and you can see those. All of these are a bacilli shape. We're going to get to the other words there in just a minute, but the shapes are cocci, bacilli, or spiral. Spiral is pretty self-explanatory. That's spiral. Okay. Now, cocci, so cocci is plural. The I at the end means it's plural. A single cocci is called a coccus. C-O-C-C-U-S. And a bacilli is plural, and a single bacilli is called a bacillus. And that's just to help you when I use those words so you know what I'm talking about. Okay, so our three major shapes that you have to remember are cocci, or coccus, bacilli, or bacillus, and spiral. Okay, those are the shape of the individual cells. Now the arrangement is how multiple cells interact with each other. How they are arranged next to each other. Okay? And we use specific prefixes to mean specific arrangements. Okay? And I'm only going to make you know a couple. So I'm going to tell you, well, a few of them. I'm going to make you know outline which ones you have to know. So the ones you have to know for arrangement is the prefix diplo. And that prefix diplo, what do you think it means? What number? 2. Di means 2. Diplo means 2. Now you can have an arrangement with any type of shape. So you can have, like you see up here, you can have a diplococci, which is when you have 2 circles attached to each other, right? Diplo and cocci. So when we use those words, a lot of times it's both the shape and the arrangement. Because cocci is the shape, diplo is the arrangement. Diplo, cocci. You can also have a diplobacilli, which is 2 bacilluses attached together. Okay? So diplo means 2. The other one that you have to know is staph or staphylo. And staphylo basically means, or I guess I can write, this means 2. This means a cluster. Okay, so a staphylococcus is a cluster of staph. You can also have a staphylobacillus, but that's not very common, so that's not even shown here. Staphylobacillus is not very common. Staphylococcus is the most common. And a lot of these, you may recognize that word if you've ever heard of a staph infection or a Staphylococcus aureus, which causes staph infections. Sometimes these shapes and arrangements are so distinctive that the organism is named after their shape and arrangement. So Staphylococcus aureus is named that because it is a Staphylococcus shape and arrangement. And that is so distinctive for that group of organisms that they named it after it. Okay, and then I think the last one that you have to know is strep or strepto, which means a chain. And again, you can have a streptococcus or streptococci, which are chains of circles, or you can have streptobacilli, which are chains of bacilli. Okay, so that's what you have to know on this slide. I know that there are other names up there. You don't have to know those. All right, so when I'm talking about bacteria and I use the words streptococcus or just coccus or bacillus or whatever else, it's important that you know what I'm talking about. So when I say something is a gram-negative bacillus, that should bring up the picture. Gram-negative is gram-negative pink or purple. Pink, gram-negative is pink, and bacillus is a rod-shaped organism. So under a microscope, a gram-negative bacillus is going to be pink and it's going to be rod-shaped. Okay, and these are just some pictures of some shapes and arrangements. These are just more examples. You don't necessarily, for this class, have to know how to identify them. But these are just some examples. So this one, are those individual, that one's kind of tricky. We'll look at like this guy down here. Or this one right here. Is that circles or like rods? Circles, that was a bit tricky because some of them look kind of ovally, but I would say that they're closer to circles. I'm not gonna, if I ever test you on anything like this, it'll be really, really obvious. This one's a bit tricky because it's a little elongated. But if I had to say one or the other, I would say circles. So if it's a circle, is it a cocci or a bacilli? It's a cocci, it's a circle. Is it gram-positive or negative? Positive, it's purple. And then what's its arrangement? Diplo. Yep, because most of them are in pairs. So you have a pair, a pair, a pair, pair, pair. And when you're looking at gram stains, this is just general advice. Always go with what you see the most. Because one of my favorite things to say is bacteria don't read textbooks. Okay, nothing is ever going to be perfect. You have to go with what you see the most. It is going to happen sometimes, like this guy right here, where 2 pairs sit next to each other and it kind of looks like a chain. So if you over-examine a gram stain, you can get a lot of different things out of this. My best advice for reading gram stains, if you're taking lab, is to read it in 10 seconds. Look at it, make a distinction, make a decision, and stick with it. Don't sit there and zoom in on every single cell and say, well, that one looks a little bit lighter. Is it pink? No. Go with what you see first. Okay, you know the difference between pink and purple, right? They taught you that in kindergarten. Just go with your gut. That's my biggest advice. Okay, this one are the individual cells, circles or rods? Circles. So are they cocci or bacilli? Cocci. Are they gram-positive or negative? Positive. What's their arrangement? Staphylo. You could argue that this is a tetrad, and it is, but we didn't use that word, so it's in clusters. Okay, this is more of a true staphylococcus, right? Those are circles that are clumped together, so that's a gram-positive staphylococcus. This is a great example of go with what you see the most, because most of this, what color does most of it look? Purple. But if you zoom in, one of them looks a little more pink, or a couple of them look a little more pink. That's just because of the stain, right? They don't read textbooks. Not everything is perfect. Most of them are purple, okay? But this is a great example of a chain. So this is a streptococcus. Okay. This is a bacillus, and this is yeah, that's a bacillus. This is a good example of not everything has an arrangement. Right? You might say that that's a diplo, and I could see that, but most of them are kind of just hanging out by themselves. So that one really doesn't have an arrangement. That's just a bacillus. Okay? So that happens sometimes. Not everything has an arrangement. Okay? That's a streptobacillus. It's a chain of rods. And then that's just a spiralia, or a spiral. And then this is another version of a spiral called, we didn't learn this word, and you don't have to know it, but it's a vibrio. It looks like a little comma, kind of like half a spiral. Okay, so those are just some pictures, some examples. And for this class, again, all you're going to have to know are those words that I talked about and wrote down on this slide. And gram-positive or gram-negative. Okay? All right, so structure of a bacterial cell. We are going to go through each of the structures individually. We're going to talk about each one kind of quickly. But I am going to highlight some specific ones. Now, something that I want to note on this slide, this is the overview of everything. This is also in your textbook. If this isn't big enough, you can find the full image in your textbook. But what I want to point out on this slide is, again, we're going to be talking about some things that are in all bacteria. All bacteria have these four things. And it just so happens that the four things that all bacteria have are also the same four things that all cells have. Okay. All bacteria have a membrane, a chromosome or DNA, ribosomes, and cytoplasm. And then the rest of this is stuff that is in some bacteria. So not all bacteria have the rest of this stuff. That's important to keep in mind when we talk about each one. Is this something that everything has? Something that none, only some. Okay? I will say that one of the more important things that we're going to talk about on this slide is this one right here. And this is a cell wall. We're going to talk about a cell wall a lot. And a cell wall, it is true that it is something found in some bacteria. But I would go as far as to say a cell wall is found in most bacteria. The vast majority of bacteria are going to have a cell wall. And the vast majority of those are going to have a cell wall made out of the same thing, which we're going to talk about in a second. Not all of them. There is a very small subset of bacteria that don't have cell walls. We'll talk about them too. But most, by far, most bacteria have a cell wall. And a cell wall is really critical to their function. Okay, but the rest of that stuff is stuff that you find in some bacteria, not all. All right, so we are going to talk about each of these structures, and we're going to talk about them from the outside in. So we're going to start talking about the external structures, the boundary structures, which are the ones that separate the inside from the outside, and then the internal. Okay, so we're going to start by talking about external structures. And external structures are things on the outside of a cell. So things like appendages, like flagella, things like that, and surface layers. Okay. So appendages. Appendages, again, are external structure. They are things that are on the outside of the cell membrane. Remember that the cell membrane is the border between the inside and the outside. So these are things that exist on the outside of the cell membrane of bacteria. And all of these things listed, if you go back and look at the chart, are all in some bacteria. Not all of these exist in all bacteria. These are all in some bacteria. It is very possible for bacteria to not have flagella. A lot of them don't. Okay? So flagella, and we're just going to talk briefly about what each of these are used for. We're not going to get into too many details. It's just going to be a brief kind of overview of what they're used for. Right, so flagella are used for motility. These are long and what we kind of call whip-like structures that whip around, like they kind of look like little tails on bacteria, and they whip around and cause the bacteria to move. Okay, these are some examples of flagella. This is a flagella right here. And again, these flagella are relatively long, and they look like long, thin, whip-like structures. The most common flagella that humans are very familiar with is the tail on a sperm. Sperm cells have flagella. That's how they swim where they need to go. But bacteria have flagella very similarly, which should kind of make sense. Remember that bacteria are individual organisms. So if an individual organism needs to move, this bacteria doesn't have legs. The only way it's going to get where it needs to go is by some kind of appendage. And so it uses little tails, like flagella, sometimes to move. Okay? Pili are long tubular structures. And they are used for something called conjugation. Now, conjugation is another word for, try to think about that word, conjugation. Another word that a lot of people are kind of familiar with is the word conjugal visit, right? Conjugal visit has to do with sex. So pili, bacteria don't have sex. Let's get that out of the way. They don't have sex. They don't reproduce sexually. However, they do something called conjugation, which is as close to sexual reproduction as bacteria will ever get. So conjugation is basically just DNA exchange. So bacteria can attach to each other with these pili, and you can kind of see, this is a picture of these two things right here are pili. And those bacteria can attach to each other with this pili, which sounds suspiciously like something else, right? So pili and DNA can travel from one bacteria to another. Again, this does not mean that they sexually reproduce. We'll define that more later. Bacteria do not have sex, but this is as close as they will ever get. Something called conjugation. Okay? Fimbriae are very short and hair-like structures that you can kind of see here. So this bacteria, all this like gray stuff on the outside, these are short little hair-like things that stick out all over the surface. Okay, this one up here, this is another picture of Fimbriae. You can see the little short little hair things. And this is used for attachment, as you can see there. So this is how bacteria grab onto each other or surfaces. Because again, remember, they don't have legs or arms or hands or anything like that. This is how they interact with their world, are through these appendages. So fimbriae are little short hair-like things that grab onto their environment. And then our last one are called nanotubules. And again, these are long, fin-like things that are used for energy exchange. So similar to pili and conjugation, nanotubes are used so bacteria can exchange energy. So they can lend energy to each other. And this is a group of bacteria that are attached via nanotubes. You can see all the little tubes going from one bacterial cell to another. Okay. Our next ones are surface coatings. So surface coatings, we have two different major types. I don't want to talk about this. Yeah, we got to talk about it. So we have two major types of surface coatings. One of them, and again, all of these are in some bacteria. Not all of them have it, only some. So the first type is called an S layer. And an S layer, all you really have to know about it is that it's a hard, thin layer of protein. Okay, it basically acts like a suit of armor. Because remember again, bacteria are single cells. They interact, their cells interact with whatever environment they're in on their own. Okay, they don't have skin and an immune system and a bloodstream and everything else to protect them from the outside world, right? Your insides, your guts, your organs, all your important stuff is packaged inside your body and it's protected from the outside by everything on the outside. Cells don't have that. All their important stuff is hanging out in the open. And so they have some things that are used to protect them, specifically things like S layers. They're hard, thin layers of protein that basically act like armor. And then another type is something called a glycocalyx. And a glycocalyx is kind of opposite of an S layer. It's a thick, slimy layer of protein and sugar. Okay? And it will often make bacteria look very slimy. So this bacteria here growing on a plate is one that produces a glycocalyx. We say that it looks mucoidy or wet or slimy because it's producing that slime layer. Okay? Now that glycocalyx is something that is very, very important to something called a biofilm. That's not visible. Okay, so a glycocalyx is something that's really important to the formation of a biofilm. And a biofilm is a community of bacteria that adhere to surfaces and work together to share resources and survive. Okay, so that doesn't make them multicellular, right? It's very similar to humans that live in communities, right? Humans survive better when they live in communities and villages. People in communities share the work, they share resources. One person grows this type of vegetable, this person grows wheat, whatever it is, right? When we distribute the work out and share resources, we're all better off. The same thing is true for bacteria. So bacteria can form things called biofilms. And these biofilms, a lot of the time, as you can see in this picture kind of right there, they form these glycocalyx layers. And that is how they stick together and form this large, slimy community. Now, a really great example of a biofilm that all of you are very familiar with is the biofilm that forms on your teeth. So when you go to bed at night, when you wake up in the morning and you run your tongue over your teeth, they feel pretty gross, right? They feel kind of slimy and nasty. If you let it go for a long time, it feels grosser and grosser and grosser. The reason for that is because the bacteria that grow in your mouth are forming biofilms overnight. They are attaching to your teeth and producing a glycocalyx layer that protects them from the outside environment. And that is why you have to, every single morning, remove the biofilm from your teeth, right? By brushing them. Okay? So that is how you remove biofilms from your teeth. You brush your teeth. Brushing your teeth is physically scraping biofilms off your teeth twice a day. And the truth of it is that if you didn't, those biofilms would become more and more and more established, and those bacteria would form bigger communities, and they'd survive better, and they'd start rotting your teeth straight out of your head, right? That's what happens when you don't brush your teeth. The bacteria take over, and they form these biofilms. And bacteria that are established biofilms are more difficult to remove and or kill than individual bacteria, right? So has anybody ever, hopefully I think most of you have probably had this experience, where you stayed over at your cousin's house or you stayed over at somebody else's house or whatever else, and you don't have a toothbrush, so you maybe find some mouthwash or something, and you rinse your mouth out with mouthwash without brushing your teeth. Does that do anything for the stuff stuck to your teeth? No. It doesn't do anything. It might make your breath smell minorly better until you can get home. But all that slimy gunk is still stuck to your teeth because that bacteria is not killed by mouthwash. The bacteria that has formed that biofilm, the only way to remove it is to physically scrape it off with a toothbrush. Rinsing it out isn't going to do anything. And so that's going to become important when we talk about certain types of infections. Again, I talked about my big huge abscess. One of the reasons that it was so bad was because I developed a biofilm inside my abscess. And the only way to get rid of it was to cut my leg open and scrub it off the inside of my tissue. As my surgeon very lovingly put it, I had to be power washed twice a week to keep that bacteria from establishing a biofilm again. Because bacteria and rinsing it with saline, or not bacteria, antibiotics, rinsing it with saline, that wasn't doing anything. It had to be physically scraped off. So bacteria that form biofilms can be incredibly, incredibly, incredibly dangerous. And a big part of that is the glycocalyx. Without that, they wouldn't be able to do that. All right? That is 12:15. That's where we will call it today.